a slightly different orientation in three dimensions, but we'll draw it like this for now. And the oxygen is partially negative. If you want to draw the dipoles, you would have something to this effect. And the idea is that you have these dipoles, and that means that if another water molecule were to cozy up with this one, that it would tend to want to orient in this manner. Okay, and in that way, then you can see these two dipoles are oriented, so that the negative end of one is oriented towards the positive end of the other. And I'm going to draw it in blue, or it doesn't matter what color you use, but there's going to be a weak interaction, a weak attraction between these two dipoles, where the positive proton of this is shared somewhat, in a very, shared in a very, not quite the sense of sharing electrons in a covalent bond, but shared in a very weak sense between its own oxygen, its own covalent system, and then this non-covalent oxygen from this neighboring. And so what we call this oxygen a hydrogen bond acceptor. And we call this hydrogen here a hydrogen bond donor. Hydrogen bond donor. And so this is basically our hydrogen bond. Okay. And it's not a bond in the traditional sense. It's a much weaker interaction. These are on the order of, say, 5 kilojoules per mole in energy relative to covalent bonds, which are much, much larger. All right, so let's think about the lab now. And polyvinyl alcohol as an example. We've talked about it already. I made it a clicker question in a previous lecture. Polyvinyl alcohol, otherwise known as PVA, is made from these groups here, this vinyl alcohol. And we talked about forming a radical. You mix this with, like, say, hydrogen peroxide, vinyl alcohol, and then one proton and one electron would come off of the vinyl alcohol group and make this radical form. And then you combine that with unradicalized vinyl alcohol, and in many steps you would polymerize. And as you polymerize, that radical is maintained until it's quenched. And so if I introduce more and more vinyl alcohol groups, then I could grow this polymer out. Sorry, people that are. Anyway, if I keep doing this, and I'll just do that by ... then I'll end up with something like this. A big polymer with a lot of hydrogen bonding groups on it, right? And you might draw this in this manner to designate that it's sort of infinite on its ends. And you can imagine if you had a couple of these that were in close proximity to one another, that with our borate compound, it could slide in between these polymer chains, and it could make hydrogen bonds. And again, I'll draw these. I'm not going to draw all possible hydrogen bonds, but if you are going to consider hydrogen bonds here, you'd want to go between donors and acceptors. And in the case, the donor is going to be the hydrogen group, a hydrogen atom on one of these alcohols, and the acceptor is going to be an oxygen, okay, which has those lone pairs, which I've abbreviated out of this drawing. So when this happens, of course you're going to have these hydrogen bonds. So we have hydrogen bonds here. And if you add multiple borates and you get lots of networks of these, and you can imagine that these two chains can be linked together by some crosslinks, and then maybe they can be linked to other chains, and then the whole solution will then sort of form a network of crosslinks. Okay, we call this essentially this is a crosslink. So these two chains are cross-linked. These two PVA chains are cross-linked together. Now these are cross-linked by non-covalent interactions, hydrogen bonding interactions, and you could do some sort of chemistry here, some covalent modification that would cross-link these two polymers together in a covalent interaction. That would be much stronger than this, but the net effect is the same. That this will affect the molecular properties of our individual polymer chains, and it'll act in aggregate in completely different ways. Okay, so yeah, this is really going to be the key crux of the next couple of labs, and your PowerPoint presentation you're going to do on this section. We're going to be thinking about, as I vary the reagents, like for example the types of polymers and the amount of crosslinker, I'm going to get different types of polymers. Now there's some general rules that you should think about, and we could get some examples from things like rubber, which everyone's played with a rubber band, I assume. And in rubber, as you add crosslinks, this rubber, you think of the rubber as having many chains like this. And they could be oriented in different directions. And essentially, there are covalent crosslinks I'll draw here in blue between all these chains. And as you add more and more of these, you increase the hardness of the firmness of the rubber. So you'll start out with a very soft. If you have very few crosslinks, you'll have a very soft polymer, and you can kind of stretch it out. It might not even want to snap back so easily. It will snap back. And then as you add more and more crosslinks, then you get a much stiffer spring, and the other thing will want to snap back more. Then you add so many crosslinks, eventually it'll be really hard to stretch. Okay, so that's the idea. As you add more and more crosslinks, you go from softer, stretchy polymer to something much firmer, much harder. And that should make sense, because you're changing the structure of this thing. With these crosslinks, you're engineering in these points of stability within this polymer network. And so if you had no crosslinks at all, and you heated the stuff up gently, then you could basically just stretch it out, okay? And it would never relax back to its original shape, because there's no links between the polymer chains. And they can just move relative to one another just fine. With the crosslinks, it sort of keeps them in a registry with one another, albeit a random sort of registry with one another. Okay, so that's the idea of rubber. And then for other the general idea for many types of plastics is that you're basically going to go from sort of a liquid with when you're in sort of the low cross-linking regime, low cross-link number, let's say, you'll go to a liquid, and then you'll progress as you add crosslinks to a gel, more crosslinks. And then you'll go to something that's very sort of stiff and rigid, something more rigid and firm. So this is sort of the general property. And the other thing to think about too is that as you add crosslinks, even as you go from liquid to jelly, you're essentially increasing the viscosity of the material. All right, so those are the ideas. So yeah, you can think about viscosity too. Okay. So at the end here, I have a few minutes and I can answer questions. I want to give you an example from biology, and to say, of course, that biological systems are made of polymers as well. And we talked about, in the first lecture, I believe, or maybe the second lecture, about the different kinds of biopolymers we have. We have polysaccharides, we have protein. Polysaccharides are made of sugar monomers, proteins are made of amino acid monomers, and DNA is made of nucleic acid monomers. And there's some other molecules that make natural rubbers and latex called isoprenes, and those are also natural biopolymers. But it turns out that even when you make proteins, these individual proteins, if a protein looks like this, and it has these different, so this is a protein, and a protein can have different, it's not a homopolymer, and I'll try to designate that in a second, but it's made up of different functional groups, and I'm drawing this very simply here, that are arranged on it. And if I just draw this sort of a binary, maybe I'll add another functional group here in green. But essentially a protein is made up of amino acids. These are amino acids, individual amino acids. And these individual amino acids will have different functional groups on them. Without getting into the details, but these actually can then assume a particular folded three-dimensional shape. Okay, so the protein actually, through hydrogen bonds and through molecular interactions between these individual amino acids, through evolution they've been selected for, so they fit like a three-dimensional jigsaw puzzle into a three-dimensional protein. And it turns out that these proteins on occasion can be selected for their ability to interact with one another. So they can make basically this type of structure, where these proteins could make a polymer of proteins, if you will. And there are non-covalent interactions, again, hydrogen bonds and hydrophobic interactions, that can hold these proteins together, okay? So there's non-covalent interactions between these. And what I've essentially tried to draw here is an example of an extracellular assembly that occurs on bacterial pathogens. And so here's a bacterium here. And on the surface of the bacterium will be these appendages called pili, okay? An individual appendage would be called a pilus. And each one of these little dots, I mean, this is not drawn to scale, are these proteins, okay? So each one of these little dots is a protein. And then you'll have the bacterium trying to infect, say, some host cell or tissue. And the way it's doing that is it essentially builds these polymers like Spider-Man, silk comes out of its arm. And they're extremely strong. They're very thin. And on the tip of them, they have features that will interact very tightly with the host cell. They recognize the host cell, and they stay attached, and they don't break. But you might say, well, how can these weak non-covalent interactions possibly achieve this? Well, it turns out that pili, or pilus, has crosslinks. And in between these subunits are actual residues. For example, this residue, and don't worry about this won't be on your exam, I'm just showing you the chemistry for people that are interested. And this is a lysine residue here, and that amine can react with a neighboring residue called asparagine. And asparagine looks something like this. Asparagine and lysine are both different functional groups in a protein polymer, but when they're close by and there's a glutamate to catalyze this reaction, then you can get a cross-linking reaction. So you can get this cross-linking reaction that looks something like this. And so now, from one protein, I've joined the other one through a cross-link, and I made this what we call an amide bond. Okay, so that's an amide bond. You might call it an isoamide bond for protein because it's not a typical amide bond that you see in the backbone. But this amide bond is going to then hold together. These cross-links. These cross-links are going to hold together these individual folded proteins and make the pilus extremely strong, while being extremely thin, and allow for that adhesive interaction to form between the bacterium and the host cell. And so that's the end. And I will entertain questions from the lecture or any questions in general. Are there any questions? Okay. Oh, question.